In this video, we're going to talk about the structure and function of lipids. And again, most of the sources of our diagrams and explanations are from our Cognitive Higher Level Biology textbook. So lipids are made of carbon, hydrogen, and oxygen, just like carbohydrates. However, their properties are very different. So they're insoluble in water, but they are soluble in other non-polar substances. They also provide 2 times the amount of energy per gram than carbohydrates or proteins. Lipids will help some vitamins to be absorbed, the fat-soluble vitamins. And then lipids also make up the cell membrane. They also provide the waxy coating on leaves and the waxy cuticle. And they also make up steroids, which form various different types of hormones. And they form cholesterol. The first type of lipid we're going to talk about are triglycerides, and they're also known as fats and oils. And they're composed of glycerol, which is a 3-carbon trialcohol. So on every carbon there's a hydroxide or an OH group. And triglycerides and the glycerol and the 3 fatty acids are combined through dehydration synthesis or condensation reactions. The bonds in this case are called ester linkages, and we can get triglycerides from our food, or they can be built by our liver. If you want to learn more about what is fat, you might want to watch the TED Ed video titled "What is Fat?" Again, here's a picture of the condensation reaction. So we have our fatty acid, and each fatty acid will form a condensation reaction with each of the hydroxyl groups on glycerol. So you'll end up with a triglyceride plus 3 water molecules as well. A saturated fat is when you have all single bonds in between the carbons in the fatty acid chain. So the molecule is said to be saturated with hydrogen. The chains are straight and attract each other through intermolecular forces. Because those 3 chains in the triglyceride are being attracted to one another, that causes the fat to be solid or semi-solid at room temperature. So the main sources of saturated fats would be animal fat, coconut oil. So some vegetable oils are also have some saturated fats, but generally animal fats is the main one. So you can see here there is no double bonds between any of the carbons in the hydrocarbon chain. So this is a saturated fat. And you can see that it is straight. In unsaturated fat, on the other hand, the fatty acids contain at least 1 double bond between the carbon atoms. So it's not saturated with hydrogen. So that's where the saturated versus unsaturated comes from. Each double bond in the chain will cause a kink, and it decreases the intermolecular forces between each chain because they can't neatly pack together. That reduced attraction will cause the fat to be liquid at room temperature. If a fat is polyunsaturated, it just means it has many double bonds in the carbon chain. So some sources of unsaturated fats would be vegetable oils and fish oil. So in this picture, there's 3 different types of fats being represented. So the first one, saturated fat or stearic acid, this one has no double bonds. This one is a monounsaturated fat. So you can see right here, after that double bond, the chain kind of bends and goes in a different direction. This example, you see the chain going into multiple different directions because it's a polyunsaturated fatty acid. So the first one's stearic acid, oleic acid, and then linolenic acid, which is an omega-3 fatty acid. Now, trans fats are a type of unsaturated fat that is mainly processed in the lab. Their hydrogen atoms are on opposite sides of the double bond, and that will straighten out the chain again and make them pack more tightly and make them solid at room temperature, just like saturated fats. So if you look here, the way you can remember cis fatty acids the double bond, the two hydrogens are on the one side, so it actually makes a kind of a C-looking structure, whereas trans fatty acids, the hydrogens are on opposite sides of the double bond. So the saturated fatty acid, no double bonds. The trans fatty acid has a double bond, but because of the reconfiguration of the double bond, it straightens out the chain. So these two aren't so great for our health. And then this one, the unsaturated cis fatty acid chain has that kink. So it reduces the London forces or the intermolecular forces, which then makes it more liquid at room temperature. So we're going to look at the energy storage for different living things. So first of all, plants will store fats for later use as well. They'll be stored in seeds. So when the seeds start to germinate, they use the fat stores for energy to grow. So seeds are basically, they bring their own food package so that they can grow for a while until they can perform photosynthesis for themselves. Endotherms, which are warm-blooded animals like us, store up fats in adipocytes to help maintain a constant internal temperature. But then when needed, fat droplets can be converted to ATP to help generate heat. Fat can also be stored as blubber in Arctic animals to act as an insulating layer and also as an energy source. Phospholipids are glycerol plus two fatty acids and a phosphate. So the two fatty acids are not polar, the phosphate and the glycerol are polar. The phospholipid has a hydrophilic head and two hydrophobic tails, which makes phospholipids amphipathic, which means that one half of it, the molecule is polar, and one half of the molecule is nonpolar. So when you add a phospholipid to water, they all form into spheres or micelles. The hydrophilic heads will dissolve in the water, and the hydrophobic tails will all aggregate and mingle in the center away from the water. And this is kind of how it would look. So you have, and this would be an entire sphere, but I'm just going to draw one ring. So the hydrophilic heads are on the outside mingling with the water, and the tails, I'm just going to draw all the circles first, it'll make it easier, and the tails are all in the inner part, protected away from the water. This is what a phospholipid looks like. So there's the phosphate, here's the glycerol, so the phosphate polar, the glycerol is kind of the in between, and then the fatty acid chains are not polar. And again, they form ester bonds as well. Phospholipids also form bilayers to form this, like for instance, our cell membranes. This is a drawing of just our simple cell membrane. So we have our hydrophilic heads on either side of the membrane. So the watery environment of the internal part of the cell and the watery environment of the external part of the cell. And then all the fatty acid tails are held in between the bilayer. So the outer parts of the bilayer are hydrophilic, but the inner parts are hydrophobic. This helps make the cell membrane selectively permeable. So the cell membrane, like I said, phospholipid bilayer. We can find water on either side of the cell, but the hydrophobic tails are protected in the center. Water can't just pass through the layer because of this. It has to pass through special water pores. Cell membranes have embedded proteins and hydrophilic pores to let the water, the ions, and other polar molecules through, which is why the membrane is selectively permeable. Now, steroids are a special type of lipid. They're nonpolar molecules that are made of 4 fused carbon rings, and they each have different functional groups. These molecules can move freely through the phospholipid bilayer, and some examples of steroids would be cholesterol, which is the simplest one. Testosterone and estrogen, or estradiol, is another name for estrogen. Cholesterol makes up part of the animal cell membranes. It's mostly hydrophobic, but it does have a hydrophilic portion, and its job is to position itself between phospholipids in the membrane, and it reduces the fluidity of the membrane and reduces its permeability to some of the hydrophilic ions. It also helps the membrane to curve, and that's what allows for the formation of vesicles or vesicles.